Enzymes are a really important part of Module 1, and this is how you can break them down. So enzymes are biological catalysts, which means they speed up chemical reactions in cells, and without them, we wouldn't be able to perform the most important cellular reactions that happen in our body. There's a lot of different things that affect the rate of enzyme activity. One of them is temperature. Every enzyme has an optimal temperature at which they perform. Below this temperature, because of the low kinetic energy associated with low temperatures, there's less enzyme substrate collisions, and so less enzyme activity. However, above the optimal temperature, we have irreversible breaks in the intermolecular bonds between the enzyme atoms, which causes irreversible denaturation, and you see a significant drop in enzyme activity. So you have this asymmetric hill. pH is another thing that affects enzyme activity. pH refers to this specific concentration of hydrogen ions in the environment of the enzyme, and this can change and alter the intermolecular forces around the enzyme, which is also resulting in denaturation. In this case, you get denaturation on either side of the optimal pH because you have equal amounts of changes on either side, so you get a nice symmetrical hill. Substrate concentration is another thing that affects enzyme activity. This refers to the concentration of the thing that binds to the enzyme. So obviously at lower concentrations, because there's less substrate for the enzyme to bind to, you have less chemical reactions and less enzyme activity. However, as we increase the substrate concentration, we get a higher and higher enzyme activity. However, until a point, which we call the saturation point, after which there's no more enzymes available for the substrates to bind to. Follow for more biology tips.